So in this recording, what we want to investigate is kind of the molecular underpinnings of development. So we keep talking about turning genes on, and different genes are on in different cells. Well, where does that come from? And so what we're going to do is go back to the single cell that you started from, that almost all eukaryotic life starts from. That is the fertilized egg, right? Or what's called the zygote. And we'll go over this when we go over reproduction. But that one cell, which has the same DNA in it that every single cell in your body has, gives rise then to all the trillions of cells that comprise a human being. And again, we've highlighted before the idea that, you have different kinds of tissue, some of it incredibly specific, but it all came from a single cell. So here's a fertilized frog egg, newly hatched tadpole will go on to develop into a frog. So some terminology that we need to be familiar with is called, one is cell differentiation, the process by which a cell becomes specialized. So again, you've heard of stem cells, and the idea is that stem cells are undifferentiated. So they're cells that have potential to still become different kinds of cells, and we're learning better and better how to direct the development of that undifferentiated cell into the cell type that we desire. So morphogenesis then is on a larger process, a larger scale, where the physical process that gives an organism its shape. And that's going to be important because what we're ultimately going to see is that the shape of an organism is going to be the result of how things are laid out in that single cell that is the egg, and how things are laid out in gradients in that egg, that single cell, are going to ultimately result in differential gene expression to all your cell types. Whew. This is a tall order, and it's in a short, small package, but at least you're going to have heard of this so we can talk about it more and think about it. But ultimately, we need to consider that egg, okay? The egg from mom. Cytoplasmic determinants are going to be key. So in that egg cell, in the cytoplasm, are RNAs, proteins, and other substances, and this is the key, and that's why it's underlined, that are distributed unevenly in the egg cytoplasm. Because when that egg divides, what you're going to get are two cells that have different transcription factors potentially in there, and that's going to result in differential gene expression. All right. Now, as cells divide, they also are positioned near other cells that are expressing different genes, and that's going to lead to what's called inductive signaling. So induction then says, I'm receiving signals from the cells around me that tell me to turn on certain genes as well. And so the best way to look at this is with pictures. So again, if you see the cytoplasmic determinants in this egg cell, this unfertilized egg, they are distributed unevenly. And when it is fertilized, that zygote then gets the signal, now that it has all its chromosomes, to start dividing. And that initial division, then you can see there are two different populations of cytoplasmic determinants in those two daughter cells. Those are going to lead to differential gene expression. And as they divide, they create cells that turn on genes that secrete different inducers that signal to the cells around them their positional location. And that location then basically sends a message to the nucleus that says, we need to turn on these genes because we're supposed to become this kind of tissue. But all of this cascading information starts with the uneven distribution of cytoplasmic determinants in the mother's egg cell. So maternal effects can be very, very strong. Now, as cells develop, they become determined, right? So again, this idea that I have a stem cell of whatever type, it has totipotency or pluripotency, the idea that it can become a different kind of cell or many different kinds of cells. Well, as we push it down the developmental pathway, it becomes determined, where it is set now in its path. And we're learning more and more how we can reverse that, but for now, let's just set a cell on its path and have it develop into the cell or tissue type that it needs to be. And so we'll look at myoblasts as an example. So MyoD is one of these master regulatory genes. These are the genes that are going to be turned on early in cell development. They're going to push it down a particular pathway, in this case to become a muscle cell. So what we can see here is that ultimately in this precursor, this embryonic precursor cell, MyoD is off. Now, when it receives information to turn on MyoD, whether that is inductive, or some cytoplasmic determinant that gets turned on later in development, ultimately the MyoD protein will be turned on. And when it turns on, then it acts as a transcription factor for another gene. And that gene product can turn on subsequent genes. So this idea of sequential regulation, we've initiated a cascade that will lead to differential gene expression that is specific to that cell. All right? So the other thing that we want to consider as we think about that egg and its gradients are establishing the whole body plan. How do we move towards morphological development, right? So pattern formation is very, very important. And the molecules that are in the egg, at what side of the egg, are going to ultimately determine the head and the tail and the sides of an organism, the back and the front. So it's positional information. They're going to establish the axes of an organism. All right. So Drosophila is an incredibly well-studied organism for development, and so we're going to look at a Drosophila example here. So when we look at a Drosophila, it's a fruit fly. We have an anterior or head end and a posterior back end, and we have a dorsal and ventral side, a back and front. And what we're going to see is that this organism has many, many, many cells, but these axes, this orientation, is actually established in the egg cell. And so we're going to see as mom develops that egg, what she's going to do is establish those gradients that one end of the egg is going to become the head, and the other end of the egg is ultimately going to lead to the cells that become the tail of the organism. And so this was a Nobel Prize-winning discovery, and what it led to is the idea of what we call homeotic genes. These are genes specific to controlling patterns of formation in early development. And so you'll hear these homeotic genes, and you'll hear what they're called also homeobox genes. Because in these master regulatory genes that are going to be turned on very early in development to establish the orientation of an organism as it develops, have this conserved sequence called a homeobox. Now, one of the mutants, a homeobox mutant, can result in drastic changes. This one's called antennapedia. And so antennapedia. So normally on the wild type, or the normal fly you see on the left, its antennae are on its head, right? Sensing the air and doing their thing. Now, pedia means foot. So this mutant in a homeobox gene actually caused legs, relatively well-developed legs, to form where the antennae were supposed to form. Mutation. So this shows you that when you get mutation high enough, kind of in the hierarchy of gene developmental control, that gene says we're supposed to be legs, and so it sends a cascading effect down to all those cells to be like, form legs, and that's what they do. And so you get legs on this fruit fly coming out of its face. So you can see that these homeobox genes are very, very powerful when it comes to development. And so one that we want to look at, one of these egg polarity genes, is actually going to be called bicoid. So bicoid is going to establish the axis longitudinally, so head and tail. And so when we look at the bicoid gene in a normal larvae, Drosophila larvae developing, it has a head and a tail. If you have a bicoid mutant where bicoid isn't working, you basically get two tails. So there's no real head formation. And what they've shown in their studies here is that actually when the egg is being developed, mom is going to lay down lots of bicoid mRNA at one end of the egg. And that will ultimately result in a gradient of bicoid, the protein, across the egg and across the early embryo, which then leads to a gradient, which an axis can then be established, right? A head end where there's more bicoid and a tail end where there's less. Right? So again, this all comes back to gradients across the egg cytoplasm. Right? So these morphogens, or this idea of these are products in the cytoplasm which are in a gradient that establish the axes and the gross morphology of an organism is very, very powerful in biology. And so bicoid is a great example because it shows you very specifically that one protein can establish pattern formation, and that mom, in the formation of the egg, mutations that she incurs that affect how she creates the egg can affect her offspring pretty dramatically.